Hey guys, this is Mrs. Allgood. Happy Wednesday. I think it's Wednesday. It should be March 18th. Okay, so before we get into life cycle of an angiosperm, I promised you yesterday I'd tell you a little bit more about strawberries. So strawberries are considered an accessory aggregate fruit. It says the many ovaries of the strawberry flower develop into achenes, which become embedded on the surface of the enlarged flower receptacle. So, bottom line is, these are your sepals. This, the part that you eat, is actually the receptacle of the flower. Okay, and remember that's the part like where all the flower parts attached. The little things that you would call seeds on the surface are called achenes, and achenes are considered a dry fruit. So, when you say, well, where's the seed? The seed is inside the achene, okay? So the achene is the actual fruit. So the little things that you think are seeds are actually the fruit, and the actual seeds are inside the achenes. The part that you eat is actually the receptacle. So, think about that next time you have a strawberry. A banana is an herb. So an herb is a plant whose stem does not contain any woody tissues. Banana trees are therefore not trees because their stems do not contain any woody tissue. They are herbs and so should be called banana herbs. It says, if a banana is a fruit, then where are its seeds? So I may have shown you this before, but this is what a banana looks like in the wild. Wild bananas have obvious black seeds. Apparently the plants of commercially grown bananas are sterile and the seeds only develop into tiny black specks. Commercially grown bananas are sterile triploids. Remember we talked about polyploidy and sympatric speciation. And then a coconut. So we mentioned yesterday that the coconut that you think of is actually the seed, okay? The coconut fruit is a drupe, okay? Not a true nut. A drupe is a fruit which has an outer fleshy part surrounding a hardened pit. So the coconut that you think of is actually the hardened pit, okay? There's your husk, which is the outer part of the fruit. So it says the white flesh and water are the insides of the seed. So, anywho, we're going to get into life cycle of an angiosperm. So you might be excited that this is the last life cycle that you have to learn about, but that means that tomorrow I think we're starting plant cells and tissues. Life cycle of an angiosperm, the only thing that's really going to make it like super different than a gymnosperm life cycle is a couple things. One, you're not dealing with, pine cones. You're dealing with flowers. And second, there's something called double fertilization that is unique to an angiosperm, and that's thought to be sort of an evolutionary advantage. And you'll see why by the end of notes. Okay? So find your life cycle. All right, we're going to be filling this in together. So I know I normally do this kind of at the end of notes, but for this, to me, it made more sense to do at the beginning, and then I can go into some more detail about specifics of this life cycle. So we're going to start up here. Okay, this is the mature flower on your sporophyte. Okay. So the sporophyte, once again, is going to be the dominant generation here. And we're talking about flowers as the reproductive structures, not pine cones. Okay? So we're going to have a diploid cell in the anther. Remember from yesterday that the anther is the male part of the flower. Well, the stamen's the male part of the flower, but the anther is that fuzzy part on top that has the pollen grains. And then a diploid cell in the ovule, because the ovule is going to be part of the carpel, which is the female part of the flower. Okay? So remember that a sporophyte is diploid. So we're starting with diploid cells here in the sporophyte. All right. So remember that sporophytes make spores, and angiosperms are also heterosporous. So they do make microspores and megaspores just like your gymnosperms did. So the diploid cell in the anther is going to make the microspore, and the diploid cell in the ovule is going to make your megaspore. Okay? Now, if you keep in mind that spores are haploid, but the sporophyte is diploid, okay, then you probably can predict what process should be occurring here. All right? That should be meiosis. Because if we're starting with something diploid and we're making something haploid, meiosis has got to be occurring there. All right? Now, those spores have got to develop into the gametophyte. So remember that the microspore is going to develop into the microgametophyte, which is the male gametophyte. So we'll tackle that one first. The microgametophyte, keep in mind, is going to be your pollen grain. And then the megaspore is going to develop into the megagametophyte, which is going to be the embryo sac. So I'll be sure to point that out to you in notes today. Now, spores are haploid. Gametophytes are also haploid. So if you're going from haploid something to haploid something, you're going to have to do mitosis. So mitosis is what's going on here. Okay? So now keep in mind what gametophytes make. Gametophytes make gametes. So the microgametophyte or the pollen grain is going to give rise to your sperm cells, and the megagametophyte or the female gametophyte is going to give rise to your egg cell. Now remember that in gymnosperms and angiosperms that we don't go straight to fertilization. We've actually got to have pollination occur first. Okay, that's where the pollen grain lands near the micropyle of the ovule, because it's going to form a pollen tube, but we'll get to that later. And then we can have fertilization. Now here's another big difference between gymnosperms and angiosperms, okay? Remember we said in gymnosperms that the length of time between pollination and fertilization was going to be something like 12 to 15 months, and we don't know why it takes that long, but it takes a long time. Okay, that is not going to be the case here. This is going to take a lot less time, probably, hours, maybe days, but not that long. All right. So once fertilization happens, we get our diploid zygote. So now we are back to a diploid something. And that zygote can develop by mitosis into an embryo, which is contained within a seed. And then by mitosis, that little embryo can develop into an entirely new sporophyte, a new angiosperm. So what I'm hoping that you see is that this really isn't that different than what we did for gymnosperms, okay? Kind of like the difference between mosses and ferns. Some of your structures are going to be different. The fact that we're talking about a flower on a sporophyte and the anther and the ovule, well, the ovule, I guess, was still the same, but the anther's a little different. And of course, that there's not a large length of time between pollination and fertilization, but other than that, this really isn't all that different. Okay, so let's get into the step-by-step here. So diploid cells in the anther, so we're starting off on the male side of things, are going to undergo meiosis to produce haploid microspores. Okay? That we took that right from our little cycle that we did. And those haploid spores are going to undergo mitosis to form the male gametophyte, because that's what spores become. Hopefully in seeing all these life cycles, that's kind of getting cemented into your brain, that those haploid spores are going to become the gametophyte. And the male gametophyte in both the gymnosperm and the angiosperm life cycle is the pollen grain. And so each pollen grain or microgametophyte has two specialized haploid cells in it. Now this is really important. Remember I told you that the one thing that's going to make the angiosperm life cycle super different from the gymnosperm life cycle is this double fertilization aspect. And so these two cells here are going to help with that. So first is called a generative cell. So these are basically the two different kinds of cells that are produced from the pollen grain. Okay, so the generative cell is going to give rise to two haploid sperm cells. So remember we said double fertilization. One sperm cell is going to do one thing and one sperm cell is going to do another thing. Okay, and then your tube cell is going to produce your pollen tube. The pollen tube is how we're going to get the sperm cells from the top of the stigma, which is that sticky surface at the top of the carpel, all the way down into the ovary where the ovules are. Okay? Let's keep going. All right, so I've got kind of a crazy picture here. Hopefully you have the same picture. If not, you may want to draw that. Okay, so this would be an ovule down in the ovary of the flower. Okay, so this entire structure is the ovule. All right. What I did here was I outlined this inner layer in yellow, and that's your embryo sac. And so you should be thinking, okay, embryo sac, embryo sac. Where did I hear that? Remember that the embryo sac is the female gametophyte. So it is that yellow thing that is outlined here. Now, within there, all right, you've got a couple different things. First, remember that the entrance to the ovule is called the micropyle. Okay, that's a term that you need to be familiar with. That is the entrance to the ovule. One of the cells that will be near the micropyle, near the entrance to the ovule, is going to be your egg cell. Okay, now I know you see all these other cells here. Those are not all egg cells. Okay, they're called different things. We don't really need to be aware of that. But I need you to know that your egg cell has got to be one of these cells here closest to the micropyle. Now, this part that I drew in the middle here, this is called the central cell in my messy handwriting there. And in the central cell, you will find two polar bodies. So you've heard of polar bodies before. Remember when we talked about meiosis, or specifically oogenesis, okay? We said that in meiosis, you make one functional egg cell, but then you make three polar bodies that die. Well, these polar bodies aren't going to die here. They're actually going to serve a pretty decent function, but they are separate from the egg cell. So you have two polar bodies contained within your central cell, and then you have this little egg cell here. Okay, so you may want to go over some of that on your drawing, or if I didn't include that picture, you draw that yourself. It's very important to me that you understand sort of the anatomy of an ovule. Okay, so now we're switching to the female side of things. In the ovule, a diploid cell is going to undergo meiosis to produce a haploid megaspore. Okay, because we're talking about what's happening within the sporophyte. Sporophytes make spores, but remember the sporophyte is diploid and spores are haploid, so that has to be meiosis occurring there. All right, then once we have that megaspore, remember that the spores become the gametophyte. So those haploid spores are going to undergo mitosis to form the cells of the female gametophyte, which is the embryo sac. One of those cells in the embryo sac, there you go right there, is going to become your egg. And then we'll have these other cells, like these other two, which are polar bodies. They're going to do something different. Okay? If I go too fast, just pause me. All right. So let's talk about pollination. All right, let's look at our picture first. So I drew over it a little bit, sorry. But when a pollen grain lands on the stigma of the carpel, okay, if they are the same species, I know this is going to bring back other stuff from evolution, but remember how we talked about like gametic isolation, and we said that, a pollen tube's only going to form if it's pollen from the same species. So if these are the same species, then when the pollen grain lands on the stigma, the pollen tube will form, and it will go all the way down the length of the style. It will wrap around, and it will join on to where the micropyle is, where the entrance to the ovule is. Now notice what that tube is for. That tube is for carrying the two sperm cells that were made by the generative cell. The pollen tube was made by the tube cell. Is that what we called it? Well, now I feel like I have to go back. Yeah, we called it a tube cell. Okay, just making sure I'm not going crazy. Okay, so pollination occurs when a pollen grain, which remember is the male gametophyte, think about your pollinators with flowers. This is usually going to be insects or birds. Lands on the stigma. And then a pollen tube grows down the style from the pollen grain really to the micropyle. I know it says to the egg there. It's going to be to the micropyle first. And then that's the entrance to the ovule. And so then that can release the sperm cells into the ovule. And then like I said, two things are going to happen because we've got two sperm. Okay? So look at your picture again here. You've got your ovule. This picture is upside down. So, if I were to do that to you, I'm not trying to trick you, but you have to look and see where the micropyle is. So this is where the pollen tube would be joining onto. The egg is always going to be one of those cells that's closest to the micropyle. And then you have your two polar bodies in this central cell in the middle. And then notice that's your embryo sac, and that is contained within the ovule. Okay, so let's look at step number 7. The pollen tube penetrates the micropyle, and it will release two sperm into the female gametophyte, which is the embryo sac. So those two sperm are important because one is going to do one job and one is going to do another job. So a tale of two sperm. Now I learned my lesson yesterday. I'm not going to click on this video clip in case it is copyrighted. So I will send you that animation. I believe it just shows you the pollen tube forming. But I am not going to risk getting copyright shamed by YouTube. So I will send you that link later. Okay, so a tale of two sperm, like a tale of two cities. One sperm nucleus is going to, and I know you say nucleus, why just the nucleus? Don't worry about that. One sperm is going to unite with the egg, forming a diploid zygote. That's what we'd expect to happen. And that diploid zygote will become the embryo, and the ovule will undergo mitosis to become a seed. So just like we said yesterday, that at the end of our gymnosperm life cycle, that we have an embryo contained within a seed. Okay, So the pollen tube's going to attach here. It's going to discharge one sperm, which will combine with that egg to form your diploid zygote, and that will end up becoming your embryo. Okay, the other sperm cell is going to fuse with this central cell right here. Okay, remember there's already 2 nuclei in there, so when we add a third one, it's going to create a triploid cell. Okay, because we have our sperm cell plus these 2 nuclei, that's going to make a triploid cell. So this triploid nucleus divides to form a tissue called endosperm, which is rich in starch and food reserves. So why that? Well, this is what we're calling double fertilization. The fact that we have 2 sperm cells, one does one thing, one does another, it's basically ensuring that you're not going to waste energy making nutrients for an embryo unless fertilization's happened. So if we have an embryo, then we're going to make the endosperm, we're going to make nutrients for it. Okay, let's talk a little bit about seeds and monocots and dicots here. So a seed is going to consist of an embryo, a food supply, which is going to be endosperm, and a seed coat. This is just a little bit more detail on the difference between the seed structure in a monocot and a dicot. So in monocots, remember these are plants having only one cotyledon or seed leaf in the seed. The endosperm develops into a large nutritive structure and occupies a large area within the seed. So what I wrote out to the side here is that endosperm and cotyledons stay separate. So this is your cotyledon. Remember in the corn, that's the little white thing that's in your corn kernel. And this is endosperm. They stay separate. Okay? In dicots, the endosperm is absorbed into the cotyledon and eventually disappears. So your cotyledon, okay, this is why it's called a dicot, because you've got 1, 2 cotyledons. This is one and that's another. And the endosperm gets absorbed within the cotyledons, so they don't stay separate. Not that that's a huge deal, but it is a difference between them. Again, if I go too fast, just pause it. Okay, why double fertilization? Why is this such a big deal? Because it basically ensures that you're not going to waste energy making nutrients if there's no embryo to nourish. So if fertilization doesn't occur, you're not going to make that endosperm. That's basically what this is saying. All right, let's finish up our cycle. We are almost done. So as the seeds develop, the ovary's wall thickens, forming the fruit that encloses the seed. So upon fertilization, remember, the ovary becomes the fruit, the ovules become the seeds. Now, again, the reason we have the fruit is to help with seed dispersal. And again, we rely on animals for that. When animals eat the fruit and then they poop out the seeds because they can't digest them, the seeds are then dispersed. And then when conditions are favorable, the seed will germinate and the embryo grows into a mature sporophyte, which is your new flowering plant. Okay, let's see if I have anything else. I don't. So a couple things, guys. I'm going to post this on It's Learning, and I'm also going to post it on Google Classroom. So if you are watching this on It's Learning, we've had a lot of glitches with It's Learning. So I sent out a code yesterday on Remind to have you join Google Classroom, because even though all the handouts and everything are listed on It's Learning, but I was concerned when I had so many students that were having trouble with it, and apparently that's because there's so many people across the nation that are trying to access that platform, and I'm not sure how well they're keeping up. So I am trying to figure out Google Classroom, and I'll still post in both places, but I wanted to have another place to post in case It's Learning was acting up. Okay, so let me end my video. I'm trying today to make you some sort of quiz that is based on last Thursday and Friday's notes for you to do on Google Forms or something like that. That'll take me a little while to make, but I'm hopefully going to have this video posted in just a little bit. All right, I miss you guys. Bye.